Hello, college biology students. We are recording today from our student naturalist classroom because animals are sweet. Now, in our cell unit, we're going to tell two major organelle stories. This is going to be the production story, how does the cell make things, and our shipping and receiving story. We're going to start with our production story first, which essentially has 4 chapters. 1, chromosomes. 2, the nucleus. 3, ribosomes. And 4, enzymes. And all of those are involved in making things for the cell, making cellular products. When we say cellular products, we're talking about things like milk, hormones, hair, oils, feathers. All of the things that bodies are made of must be made by cells because that's the functional component of the bodies. So any cellular product has to go through these stages. So let's start with Chapter 1, chromosomes. Chromosomes are made of 2 parts. The major part, the primary part, the part that is often made synonymous with chromosomes is DNA, our nucleic acid. And each chromosome is one very, very long strand of DNA that is contains 50 to 250 million nucleotides. At least those are the ranges in humans. Chromosomes are not just DNA, however. There is another component. Histones. Histones are a very small protein that the DNA wraps around. This is necessary because DNA is an exceptionally long molecule. It is some of these chromosomes, I'm sorry. Let me be clear. If you take all of the chromosomes in a cell and stretch them out, you're going to get 2 meters of DNA. Well, that is a long length to cram into this tiny microscopic space. So how do we do that? Well, the DNA coils up, but to coil up efficiently and effectively, it wraps around these histone proteins to do that. So these histone proteins help us pack that DNA in that very small space in these structures called chromosomes. Now, the functional part of chromosomes, the functional part of DNA, which, as we know, are the instructions to build proteins, are genes. And a gene is the instruction to build one protein. So, human bodies are constructed of a range of somewhere between 20,000 to 25,000 different proteins, which means amongst our chromosomes, we need to have 20,000 to about 25,000 different genes. Each species has its own chromosomal number. Humans happen to have 46, but other organisms have different numbers, and that number can range and vary widely. Fruit flies have 8, and some plants have hundreds. So those 25,000 proteins, those 25,000 genes are divided amongst those 46 chromosomes, which means there are hundreds of chromosomes on each of those genes. Not every cell in the body needs to use all of those genes. My eyes don't make stomach acid, so they don't need the enzymes that construct that acid. My tongue doesn't interpret light and send a message to my brain, so it doesn't need proteins that are light sensitive. So how do we regulate that information? Well, DNA genes that are not needed, those sections of the chromosomes are tightly coiled around the histones. The genes that are needed by a cell, those specific genes uncoil and are available to be read. So that is how our different cells in different sections of the body are able to do different things, even though they all have the same chromosomes. So, quick edit. Sorry about the disruption. Our chromosomes do contain genes, but they are not only genes. There are sections of the chromosome that are not genes, and the distinction between those two are called coding regions, which contain the genes, and non-coding regions. And these are scattered throughout the chromosome. So it's not like we've got this big block of coding section and then this block of non-coding. No, you'll have a strip of coding and then non-coding and coding and non-coding. So they'll be interspersed throughout the entire chromosome. And what's remarkable is that only 2% of our chromosomes are actually coding regions. It is 98% that is non-coding region. So if only 2% of our chromosomes are genes, what's the other 98%? That is apparently not doing anything. Well, this is actually an area of intense study. We are still studying these regions to determine if there's any significance to them. Current ideas about where they came from is that they are, one, ancient deactivated traits, instructions for proteins that human bodies no longer make. We somehow lost the ability to make those proteins, and now the relic of those instructions are just left there in the chromosomes, and it keeps getting copied every time our cells reproduce. That's one. The second is that viruses can sometimes insert their DNA into their host. Well, if they do that into a host cell, but it doesn't cause a problem, and the cell can continue to function, then the cell's going to continue to replicate that DNA when it self-replicates. And so, again, something that happened to our ancestors, we continue carrying that DNA legacy on. I hope this helps. Struggle well.